Anthony Nolan registry with over 900,000 volunteer donors willing to donate their stem cells for any patient that needs them. And there are over 41 million donors in the world, or in 75 registries worldwide. And we match our patients with their potential donors by performing HLA typing. So HLA stands for the human leukocyte antigen, which is a cluster of genes situated on the short arm of chromosome 6. So HLA genes are amongst the most polymorphic genes in the human genome and play a critical role in host defense. High-resolution HLA matching has been shown to improve transplant outcome, and next-generation sequencing is allowing us to look at up to 11 classical HLA loci.
So let's look at our sample workflow and how we automate NGS-based HLA typing. So our workflow is split into two distinct areas. We've got pre-PCR, where samples are received, DNA is extracted, and HLA loci are amplified. And then our post-PCR areas, where the samples are quality controlled, library preparation, and then sequencing is carried out. So we have one Hamilton star in our pre-PCR area, and the second one in post-PCR. So once DNA has been extracted, we can move on to amplification. So we use the Gendex NGS-Go AmpXV2 kit for amplification of 11 HLA loci. So we use a singleplex HLA primer setup, with the exception of DRB1 and DRB3, which is multiplexed, meaning that each sample, if it has all loci tested, will have 10 PCR setups per sample. So using automation, we can test up to 95 samples per batch and also 1 to 11 loci per batch. So here's a photo of the Hamilton star that we use for our PCR amplification setup. 1 and 2 here over on the left-hand side is the tip stackers. So we've got filter tips and non-filter tips for the different pipetting requirements. Number 3 is the cooling carriers. So these modules here are cooled by under external refrigeration units to a temperature of 5 degrees. Which essentially is the same as setting up your PCR reaction on a cold block as you would do manually. So plates are stored here during the setup. The black tube rack you've got here is for master mix tubes and master mix components. So the number 4 is the EasyCode scanner. So the EasyCode scanner is over here on the right hand side, and it scans the 2D barcodes on the bottom of our DNA tubes. And then compares this with the Lavas generated work list. So this ensures that all samples are in the right position and the DNA block is correct. So the core gripper, number 5, is used for moving labware around the deck. And number 6 is the barcode scanner. So this is used to ensure plates are loaded in the correct position. And then Venus software logs plate barcodes in the CSV output files. So numbers 7 and 8 here at the top are the 1 mil core channels and the multi-probe 96 head. So the multi-probe 96 head, as you can expect, is used for high throughput sample processing, and the channels are used for individual pipetting, preparing master mix. As you might notice, we do not have a thermal cycler on the Hamilton deck. So once the PCR plate setup is finished, we remove the plates and load them onto a 96 well plate thermal cycler for amplification. So once PCR is complete, we verify that amplification has been successful using gel electrophoresis. So this identifies any failures at an early stage, and these samples can be re-amplified if necessary. So we set up samples for gel electrophoresis using the Hamilton ML star in our post-PCR area. So for each amplified loci, selected samples are quantified to get an average concentration. So this concentration is then used in an equation with the amplicon size to pool together different HLA loci tested for each sample. So pooling is also carried out on the Hamilton ML star with the same deck layout you can see here on the left. So once HLA loci have been pooled, we can commence with NGS library preparation. So we use the Gendex NGS-Go kit, which follows a traditional NGS workflow. Fragmentation of amplicons followed by ligation of adapters, a bead-based cleanup, and then addition of unique indexes allows us to create a barcoded library of DNA samples that can be pulled together and loaded on the Illumina iSeq or MySeq. So we perform end-to-end NGS library preparation using our post-PCR Hamilton ML star. And as you can see, it's slightly different from our pre-PCR robot, but only slightly. So the main difference here is our multiplex carrier, number 3 here, which has a number of different modules attached to it. So the module at the front is a reagent trough carrier, which allows us to load large volumes of reagents. The magnetic plate adapter just behind is very useful for bead-based cleanups. And then right at the back, just behind here, you can't quite see it, is the Hamilton heater shaker, which we use for plate shaking. So the versatility of this multiplex carrier is really helpful for performing NGS library preparation.